All right. Hello, everyone. Good evening, and welcome to the third edition of Tech and Health. And we am so sorry for starting very late today. I mean, it's been a blessed day, but it's such a great one that finally we are here. A lot of things happened, and it's almost like this edition was not going to happen. But yes, here we are with the edition happening. So because we are far behind and our time is fast spent, we are just going to be getting right into it. And I wanted to say, give a recap of some things we did in the previous edition, but probably if we have more time, I might drop. Okay, I will drop the link to the previous video in the description of this video. So for our new subscribers and if you are joining us for the first time today, just check the description and click on the link for the previous video so that you can know what we are doing on this show called Tech in Health. And so without further ado, I'm going to be inviting our guest to join us on stage in person of Dr. Emeka Umezurike. He's going to be introducing himself by himself, right? So good evening, sir. So sorry for joining late.
Yeah, hello, good evening. I hope you can hear me well.
Yes, sir, I can hear you well, sir.
All right.
How is network on your side?
Yeah, it looks good. It's okay.
Mm, okay. So, sir, just a little. Can you just tell us a little about your textbook that you just wrote? I think it's about the foundation of microbiology.
Yes, thank you very much. I'm happy to
I
be
think
here.
you
My name
lost
is
me there.
Dr.
All right. Basically, for microbiology students. So, can you just tell us a little so our audience can. Sir, can you hear me now? I think there was a
Yeah, I can
network
hear you.
glitch there.
I can hear
All right, so I was saying that you should tell us a little bit about your textbook that you just wrote for microbiology students.
Yes, I said my name is Dr. Emeka Umezurike. I'm a microbiologist and a public health researcher. So, to the textbook, I just wrote a textbook recently on pathogenic microorganisms, introduction to medical microbiology and pathogenic microbiology. So the textbook is basically, diving into the world of microorganisms as it affects human health. So our focus in that textbook was on microorganisms that have implication for human health. So by extension, we're saying microorganisms that are pathogenic in nature that can cause one disease or the other. So we took our time to go through quite a number of microorganisms, bacteria, viruses, fungi, the ones that cause harm to humans, the ones that are able to cause one disease or the other to humans. And then we looked at their structure, we looked at how they're able to cause these diseases, and in general. So the textbook is an introduction into medical and pathogenic microbiology so that anyone who reads the textbook is expected to have basic knowledge of microorganisms that can cause disease to human beings and how these microorganisms function.
Mm, okay, sir. So leveraging on what you just said now. So for a student starting out in microbiology research, what are the top three tech skills you think this student should have, whether software or hardware, and it doesn't matter, they could be undergraduate or postgraduate students. What are the three tech skills you think is essential for them to survive in this age of tech and AI?
Yes, in this age of tech and AI, a student who is starting out as a microbiologist, and I'd like to stay by extension, say, a life scientist. What we do in microbiology is a section of life science. So when you talk
Yes, sir.
about molecular cell biology, genomics, biochemistry, microbiology, all of them together are under the life sciences. So for any student who is going to be, starting out in this area, technology has moved really far that for such a student, there are some things that the student is expected to try and learn to be able to keep up. The interesting thing about the world we live in is that it is evolving very quickly. So, and it's both interesting and sad. And why I say it's interesting and sad is because what you know can become obsolete overnight. So, but
Yes,
to answer your
sir.
question, John, for a student who is starting out, the basics should be on one, gene sequencing. That is the basics. He should learn any of the tools that he can use to understand gene sequencing, know how to do your blast, for an example, on your NCBI database, very important. Then know how to do your phylogenetic trees on your mega software, very, very important. So these are the basics. And then depending on how much interest you have, like you yourself, that I know is a very good data scientist, if you're also interested in Python and R, these are the basic things that a microbiologist of today has to get to know, because interestingly, science has moved past just culturing microorganisms in the lab. That is now at the basic level. So to be at a level where you are functional and you're able to, talk with other scientists around the world, your, like I said before, doing your blasts very important, your sequence alignments very important, being able to draw your phylogenetic trees very important, and then
Mhm.
being able to do your gene sequencing. These are very important skills that students need to know and understand.
Okay, so sir, you mentioned something about bacterial culturing earlier. I remember when I was still an undergraduate, we had to do a course, my MCB 201 or 202, and the basics of these was just culturing uh, bacteria, how to culture bacteria, culture media, and the likes. So now my question is it that these things are not as relevant as they used to be anymore, or they are just like the basics that is expected for any life sciences student to know or be aware of before they even proceed to owning these other top three test skills we just talked about?
Yes, thank you very much. Maybe my answer might be a bit biased because I grew up learning microbiology by learning to do cultures first. So maybe if you ask a younger microbiologist, he might have a different answer. But for me, um, I think that you should, as a microbiology, understand, as a life scientist, self, like I said, it's not just microbiology. As a life scientist, you should understand the basics of culturing and knowing how to do these cultures. You should be able to carry them out because they are not that difficult. But like you said, that is just the basics. You should understand them. But the thing is that tech on its own opened a totally new world to us as microbiologists and life scientists. Tech made us to understand that there are millions of what we call non-culturable organisms. So we began to
Oh.
realize that there are so many organisms that you cannot culture. And the question many would like to ask is, why can't you culture these microorganisms? You can't culture them because currently the culture media that is available to us and the conditions that we're able to simulate in the laboratories are not sufficient enough to culture these microorganisms. Now, it is more interesting to note that many years before now, we did not know of the existence of these non-culturable microorganisms. We
Mhm.
obviously thought they did not exist.
Wow.
But when next sequencing came, DNA extraction and all of that, we now realize that you can take a sample of soil, you can take a sample of fecal matter, you can take a sample of an aliquot of water or any fluid, and then run
Mhm.
analysis on it without even culturing. And all you are doing from that sample is harvesting or cultivating all the DNA that is present in that sample. So you're not
Wow.
putting that sample on any culture media. You're just simply using your extraction kits to extract all the DNA present. And because you know that every organism, whether it can be cultured or is non-culturable, has DNA. So
DNA,
meaning that
yes, sir.
if you can harvest all the DNA present in a sample, then you're able to identify all the organisms present in the sample, whether you're able to culture them out or not. So it was like when you think of the days when Anton van Leeuwenhoek, did his microscope, his very crude microscope, and all of a sudden he realized that there's a world beneath the one that we can see with our naked eyes. That is the
Yes,
way it is too. Immediately we're able to understand that wow, you can get DNA and use DNA to, identify organisms. We saw
identify the
that
species.
there's even a much larger world that we don't know about because of the limitations that culturing microorganisms has. So, a life scientist, you should understand what culturing is. You should be able to run it, but you should also understand that science has moved past that level when it comes to identifying microorganisms that are present in any sample.
Okay, so this is a digression because this is not part of the question I want to ask you. Just something I observed recently, because I'm not a microbiologist by background. So I was having a conversation with a lab technician, and it was telling me about how we need to culture bacteria, and in 24 hours we are going to know if it is gram negative or gram positive. And for me, I felt like that was it, because I know that when once we culture fungi, once it grows within 72 hours, we're able to say, oh, this fungi belongs to this group, it belongs to
Yes,
that group. And the lab technician just went ahead to tell me that no, that after culturing, we are just able to identify if it is gram negative or gram
yes.
positive, not that
Mhm.
we are able to identify it down to the species level. Then it got me wondering, so
Yes.
why are we culturing in the first place if I'm not able to identify what I'm supposed to identify? So why am I going through the stress of culturing? So I want to ask you, because let's say in the past 5 years, when the well, DNA extraction and all these things has been around for a while in the Western world. So let's say in
Yes.
African continent, just really got
Mhm.
into the world of genomic, metagenomics, DNA sequencing and the likes in, let's say, 10,
Yes.
5 years ago. So what was it like
Mhm.
in the past 5 years when you were an undergraduate or when you were doing your masters, identifying this species just at culturing level without going further, doing DNA extraction? I mean, it's going to be very tedious because you are
Yes.
just able to tell if it is gram negative or positive. You are not even able to tell if it is, sorry, I don't want to pronounce the name so I don't make a mistake. You are not even able
Yeah.
to tell if it is this or that. So what was it like then for you, sir?
Yeah, you're right. You're very right. And apart from the rigors of struggling to try to, and this applies very much to bacteria. So, apart from the rigors of struggling to try to understand the bacteria, what was even more important to note was that we had incomplete information a lot of the time. Let me give you a simple
Mm,
example. When I did BSc and then I was coming up to do MSc. But my MSc, I did it in public health, not in microbiology. But I came back to do the PhD in microbiology. So what we noticed when I did my first degree, we are doing purely culturing and all of that. Now, why I said it gives us incomplete information is that at that time, for an example, if you take fecal matter 20 years ago in Africa here, I'm not talking about abroad now. In Africa here, we believed that Escherichia coli and maybe the Enterococcus faecalis species were the most abundant organisms in fecal matter. Now, how did we
mm-hmm.
reach that conclusion? We reached that conclusion simply by culturing. And actually, what was happening was that those organisms were the ones that grew most out of those samples based on culture, not that they were the
Mm-hmm.
most abundant in those samples. So
In
when
those
you
samples.
understand, so when we started doing DNA work, we started doing molecular identification, we began to realize that E. coli and Enterococcus, they were even the least abundant. We had
Wow.
many, the Bacteroides, we had Bifidobacterium, those ones were in much larger quantities and amounts than Escherichia coli, and Enterococcus. So the point being that we had very incomplete information when we were just doing culture. Because you think of it like this: once you are trying to culture out of a sample, let's say it's water sample, let's say it's a fruit, whatever it is, when you're trying to culture, you must put at the back of your mind that whatever is going to grow is what your media has the capacity to grow.
To grow.
So
Wow.
you get, so all the other things that your media is not sufficient for, they will not grow, which does not mean that they are not present, but the conditions are not favorable for their growth.
Favorable for them
So
to grow.
you understand? So that's why when we now started doing molecular identification, we realized that we don't need to grow anything. We just need to get all the DNA that is present in the sample and then identify all that DNA, and we're able to identify so many organisms. So it was tedious for sure, but it was even more importantly, it was giving us information that was incomplete. Now we have a better picture of what is present in whatever sample that we take to the laboratories.
Okay, we are back. It was my network. Sir, can you still hear
No
me?
problem. Yeah, loud and clear. Loud and clear. I can hear you.
Mm, there's a bit of network glitch here and
Okay,
there.
but I
Okay.
can hear you. I can hear you very well.
All right, sir. All right, sir. So, sir, I another thing I wanted to ask is about the automation and the manual skills. For culturing bacteria now, because it's something manual that we teach in the lab, it's almost like it's automated. You get your media, your nutrient agar, you culture. Once it grow within 24 hours, if there are colonies of bacteria on a particular plate, you subculture, and you might go on and go on like that till you are done separating as much colony as what you find on that plate. So my question is, does the increase in lab automation takes away from the tradition? So when I mean lab automation now, I'm talking about the fact that we, like you mentioned earlier, you get the sample, you sequence, you do all those things, and you get your results. So does it take away that traditional basis that you must train in knowing how to culture? You must be able to look at a place like this and say, because it is gram negative, then it is E. coli, then it is this. Does it take away that heart of tradition, or does it just freely allow
Okay, can you hear me?
Yes,
Can
I
you hear
can
me?
hear you, sir.
All right.
Yes, sir.
Yes. Yes. If I get your question well, you're asking that the new methods, the molecular methods, do they take away, the traditional way of doing things in the lab, culturing, and all of that? I think
Yes, yes.
what the new methods did basically is that they replaced the old methods with different methods. Now when you do
Oh,
molecular identification, there are things that you have to learn. You have to learn, for an example, you have to learn pipetting, which of course we used to pipette before, but we don't do it at this level of precision. And you're also handling reagents that are quite expensive and that are usually in minute quantity. So these are new skills that you need to learn. So I think that what just happened with molecular identification and genomics is that it brought to the laboratory new set of skills that you have to learn as a scientist,
Mm,
running your electrophoresis, for an example, viewing your gel electrophoresis. And then, like, we are talking tech too. So, there are new skills that you need to learn. You need to learn the softwares. Some of us, like yourself, have to go as far as learning coding and all of that. So, before, these were skills. For an example, coding was a skill basically for computer scientists or people in
computer
software.
scientist.
Exactly. It was in my time, 10, 15 years ago, it would be strange that a life scientist is learning to code. So what I just know is that there are new skills that we have to learn. We have to learn.
Mm-hmm.
When I was doing my research before coming up for this program, I saw the fact that if a computer scientist has to do something for you, in life sciences. He can do his best, but he can't be like you, who is a life scientist, because you have basic understanding of these things. You understand the concepts, and the concepts are important before you now put tech to the concepts. So, to answer your question, I think that the old skills are there, but there are new skills. The skills have evolved, and to keep up as a scientist in life science, you need to learn these new skills and be able to carry out these new skills in science. Can you hear me? Can you hear me?
Not so clear. I think the
Okay.
network is
All right. So
breaking.
I'm done with my answer. I said there are new skills which are different from the older skills, and you need to learn these newer skills.
Yes, I can hear you now.
Yeah, so you need to learn these newer skills. It's really important. Yes, thank you.
I think we are back now.
Okay, I can hear you.
Yes,
I
sir.
can
I
hear
think we
you.
are back now. Yes, the network, I believe that. All
All
right.
right.
Okay, sir. Sir, can you take that part again? Because I'm not sure it was
Okay, I was
breaking.
Okay, I was saying that the thing is that there are newer skills to learn. The older skills are there
All right, sir.
and can be picked up easily, but that there are newer skills because of the direction in which science has gone. So you have skills in bioinformatics, you have skills in data science, data analysis, and I said that you may even need to go as far as learning to code, and you need to learn Python, you need to learn how to use R. All of these are the new skills that are expected of the modern day life scientists, which is not what it used to be before. But of course, we also need to be evolving as scientists and keep up. We can't just fold our hands. We need to keep up and not let this trend, pass us by. We need to be there and learn how to do these things so that we'll be relevant in these times that we live in.
Okay, sir. So, sir, speaking of trend, right? So there was a conversation going on Instagram. I was just scrolling to Instagram, then I saw a conversation where a group of tech guys were talking about that the highest of human invention, as it at least as at now, is the use of AI, because you can see AI automation in everything, healthcare, finance, every aspect. So now with AI now in the world, what role do you think AI is playing currently in predicting microbial behavior, especially with the rise of antibiotic resistance to viruses, to microbes, to pathogens generally. What role do you think artificial intelligence is playing in predicting the behavior of microbes, maybe in disease or endemic microbes? What role do you think it's playing basically? What role is AI playing, sir?
Yeah, AI, machine learning, because I like to think of them as brothers, AI and machine learning. They are playing a lot of roles in science. And I agree with the conversation that says that AI and machine learning, they are some of perhaps the greatest invention of man to date, maybe rivaling the coming of the internet. I personally feel that the coming of the internet was man's greatest invention. So the thing is, science is evolving a lot and is evolving really fast. And to get down to your question, when you say what role is AI playing, AI is playing a lot of roles, and those roles center around data sets, large data sets. AI is able to, now a lot of scientists are training a lot of machines, with very large data sets to be able to do quite a number of things. Let me give you a simple example. I attended an interview sometime last month, and they were talking about the fact that even our simple microscopy now, when you do microscopy as a microbiologist or a life scientist, you have what we call a micrograph. So that's a picture of what it is you are viewing on the microscope.
Yes,
Now,
sir.
AI and machine learning are moving in the direction of being able to train computer models, with millions of pictures. And what's the idea behind that? The idea behind that is that it's already been done. It's not an issue of near future. The idea is now that you can just take a picture of a microorganism. Maybe I get a microorganism in Ibadan, and then I'm able to put it on the microscope and take a clear picture, a micrograph of that microorganism, and I just feed it into these databases. Because these machines have been trained with millions of images, immediately it throws out the identity. It immediately tells me the identity of this organism. And these are groundbreaking things, because number one, you realize that that is even faster than doing your DNA extraction and your molecular
Extraction, yes.
identification. You get, it's even way faster than doing your culturing. So it's just an
Mm-hmm.
issue of the computer models have been trained with millions of pictures. So once you bring your own picture, immediately it identifies for you with maybe 98, 99% clarity or similarity, telling you that this is the identity of the organism. So that is one way that AI is revolutionizing science, because you imagine that now it means that you can go to the hospital, and by the time this thing is fully in play, it means that you can get the identity of your organism in seconds. All you need to do is maybe grow it overnight, because even the sequencing method, although you have the real-time sequencing too, which is really fast. But let's say you are in an area where resources are not as much as you being able to do the molecular identification due to cost. That means you can also go the route of just having the picture and uploading it to a database, and within seconds or minutes it gives you the identity of that organism. So these are important things. So that is one thing. Then AI is doing a lot. Many when you have your data sets, let's say you do your sequencing too. Now AI is able to look through your sequences and tell you very quickly the genes that are present in all these
At
your sequences.
present.
When you do something like whole genome sequencing, part of the really difficult work there is now trying to identify all the genes that are present,
Yes,
in
yes,
that
yes.
your whole genome. So, but now AI is working so well that, due to the fact that it already has large data sets that is working, and that's the beauty of it all. You train these models with really large data sets, such that once you bring your own data set, very quickly it is able to run through it and is able to tell you that, oh, this is similar to what I've already seen, and this is the name of your organism, these are the genes that are present in your organism. So identification of organisms, identification of genes. Then even yesterday we had a lecture at the Faculty of Science, our faculty lecture, and we were discussing so many things, even to the essence of this our antimicrobial resistance, this our drug discovery, drug all this work we
Drug
do on
discovery.
new, yeah, exactly, drug discovery. So you see that AI is doing a lot, because what AI is doing is that it's telling us, number one, where the problems are, why these drugs are unable to work. It's telling us at the genetic level, at the molecular level, what the issues are. And then even your AI is, when you have your different chemical agents, it's the chemical agents that are the drugs, and they have different
Yes,
functionalities.
sir.
Now, when the AI knows the different chemical agents and their functionalities, what they can do, and it also knows the places where the problems are, what is making these your microorganisms resistant to all these your chemical agents. It knows, which one to substitute for what. So it is giving us
For the
new
other.
ideas. You understand? Which is giving us new ideas of drugs that can be used. Although there are issues around toxicity, there are many issues. So it's not as if an AI tells you, for an example, that so drug can cure cancer. It's not as if we rush and start implementing it. Of course, we will still go through the clinical trials and all of that. But the good thing is that it's shortening the time that it takes, because it is dealing with large data sets, and it can see and analyze far quickly than the human mind can. So these are some of the things that it is doing for us in science, amongst many other
All
things.
right, sir. That was so comprehensive. So now to the fear that some people usually have concerning AI, that AI is here to, take their jobs or something. So now bringing it down to research, sir. When we talk about AI in research, a concern is in the place of interpretation of this data, because as much as we are feeding AI with data and it is giving us an output of what we want, there is also a place of interpretation. So my one question is, how are we still able to found novel novel microorganisms? So in case whereby we take a data set, so let's say we've already done DNA extraction, we want to identify these things, we've blasted it, it brings the sequence, or the sequence cannot even be found on blast NCBI, then we are trying to use AI to determine if is that this thing has been done before but NCBI database does not have it, and it's the outcome is that it is a novel research. What role does How are we able to tell it apart in that sense that, okay, oh, AI is saying this thing is novel, and because it is novel, how are we able to tell it as scientists whereby we don't cross that blurry line of, okay, is what AI says it is not, there's no room for doubting AI is always right, right? So there's
I
no room for
understand what
doubt.
Yes, you're asking around the issues of ethics. So if AI says it is A, do we then swallow it and say, oh, it is A because AI has said it? These are important questions that you're asking, because it talks to the issue of ethical limits. Yesterday, I keep referring
Mm,
back to the lecture we had yesterday because it was a beautiful lecture. The man was Professor Onifade from UI, Computer Science Department. He had all of us spellbound when he was talking a lot about machine learning. He was saying things like, okay, we know that the AI works, it's churning out answers. We know it's working with algorithms and then computational models. He said, but can you truly, as a scientist, can you truly explain in details how it is coming up with those answers? You say, okay, yeah, we know it is crawling around the web and pitting the information against whatever is out there. How are you sure that the process is without mistakes? Because the process also sometimes, like we know from this our common chat GPT, that it also generates very funny and silly answers sometimes, especially if you push it
mm-hmm.
into uncharted territories. It begins to really, really fumble. So I agree with your question, and I'll just say that the with great power comes great responsibility. That's how I look at it.
Mm,
Those who are scientists that are building these models, they must make sure that these models are built in a way that they are interrogated to the highest level. And what do I mean by
mm-hmm.
that? It would help us really if we can depend on AI and say, okay, if it gives us an answer, we are sure that that is the answer. But that means that the developer must do it in such a way that the AI model itself runs extensive research on whatever it is doing. You know, like the example you gave us, we have a set of sequences now, and we run it through NCBI. We cannot see any match, and then we take it into our AI. We ask AI to run it for us through different databases, and it also comes up with no similarity, no match. And then, so can we now say that, oh, I've gotten a new organism? I think that if the scientists, make sure that this model is really doing what it's supposed to do, we will be able to take it that yes, it means that we have been able to find a new organism. But also, as a scientist, I'll say that we should proceed with caution. Anything, I always tell people something about science because I'm a scientist and I'm a believer. And over a believer, a Christian believer. So now, over a period of time, if you know, can you hear me? Can you hear me? Hello? Can you hear me? Can you hear me?
Yes, sir. Yes, sir.
Okay.
Yes,
Over
sir.
a period of time, what I've noticed about science is that science reverses itself. And that's why I tell people that anything that has to do with science, if you've been a scientist for long, at least I have been a scientist for maybe close to 20 years now, I can tell you one thing, that science reverses itself. Theories change, you
Mm.
get conclusions and
Yes, sir.
inferences over time. So that is why, as a scientist, it's like a business where you never say never. And that means that when you take current conclusions, you take them with a pinch of salt. You realize that new evidence can arise tomorrow that rubbishes what we are holding dear today. That's the way science.
Mm.
So it's important that even with our AI, our machine learning, we are also cautious even as we proceed, because we know that if science has not taught us anything, it has taught us the fact that science reverses itself a lot. And things that you thought were good 5 years ago or 10 years ago, you find out that you are not okay with it now. So we must proceed with caution. But of course, we must make sure that these models are well, built, and they do their analysis very, very well. This is important.
Okay, sir. So, I want to ask this for upcoming researcher like myself. So, what is the biggest mistake young professional makes when trying to pivot into tech-driven biological research? Because in the age of tech, everyone wants to learn data analysis. I just want to analyze data. And I've seen so many people pivot into that, and from there, they lose their way. And the next thing you hear is science. I'm not interested. I just want to stay with data analysis. So, what was
Mm.
one of those biggest mistakes you think scientists, scientists make when they are trying to pivot into tech-driven research, biological research, sir?
Yes, some of the mistakes that a young scientist could make is the fact that the young scientist can, like you said, lose his way very quickly. I think that it is important. As a lecturer, a lot of students walk up to me and they say things like, "Sir, what are we going to do when we graduate? What do we do with ourselves? What do we do with this degree?" And it's important, I think, that every scientist sits down and asks himself, what do I really want to do? What do I want to achieve? Yes, these tools
Mm.
are there and we need to learn these tools, but you need to ask yourself, what to what end? I think that is the major question. To what end am I doing this? Am I don't want to get swept away and carried away. I become purely a data analyst. And for an example now, where if you start making a little money from doing data analysis, if you are not careful, you might totally become uninterested in even the science itself, and all you want to do is just run analysis for people
Because
and make
science does not pay on time.
make money and forget about it. But I think that as a life scientist, you must be focused. And it's important that you ask yourself, what do I want to do with science? It's important. And there are a range of many things you can do with science. We were in Nigeria now, we are really looking at entrepreneurship very seriously. So as a scientist, you need to ask yourself, what are the entrepreneurial parts of science? Which one am I interested in? Am I interested in academia? Am I interested in having my own lab and then running my own lab? Am I interested in? So these are all the different questions. What do I want to actually do with this science? Do I want to work with government? Do I want to work with WHO? Do I want to work with NGOs? These are the questions that you should ask yourself. So that as you pick up these tools, you are not confused. These tools are a means to an end. They are not the end itself. So as you pick up these tools, you learn R, you learn Python, you learn to blast, you use Mega, you use other bioinformatics software, KIME and all of that. The important thing is that you're asking yourself that this knowledge is to what end? These tools are just a means to an end. They are not the end. So you must ask yourself, what do I want to do? Do I want to be in government? Do I want to be in academia? Do I want to work with policymakers? Do I want to work with charity organizations? Do I want to work with the UN and its agencies? Do I want to be in academia? These are the questions you ask yourself. Do I want to be in entrepreneurship? You know, for an example, even the market for entrepreneurship is opening a lot in Nigeria, because think about it. You know how
Mm,
many thousand, hundreds, don't let's say thousands, now hundreds of higher institutions there are, and then the hundreds of, so you have thousands of scientists in different universities, and everybody is going towards the area of molecular work. So do you want to do entrepreneurship, getting reagent, getting kits, getting
mm.
strips, RDT kits and all of that. So these are all different areas. You need to ask yourself, what do I want to do? Chart a course for yourself and then follow it. So that's the mistake. Don't learn these tools are not they are not an end in themselves. They are a means to an end. So that, I think, is the
And
mistake
yeah.
I see a lot.
All right, so this is not part of what I intend to ask, but I just want to ask this, and that's I think I'll make it a two-in-one question. So first of all, is the place of volunteering and internship. So for a student who has identified what they want to do and they realize that they are interested in research, what advice will you give? Because right now on internet, you hear people talk about they don't want to intern without being paid, and man, nobody wants to pay an intern in Nigeria as of now, at least. Because I know of some companies that you even have to pay them to intern with them. It's that
Yes.
serious. So, what advice will you give to a student who wants to go out there to intern? Are you going to say, what advice? Sir? Don't let me give examples. Because, and there are funny people who will go as far as saying, if they're not paying this amount, I'm not interning. And I for me, I believe internship and volunteer plays a huge role in that career. Even maybe you just ended school and you want to go for NYSC, if you are able to intern between that period of time, it's just there's a way it just helps you redirect your focus into what exactly you want to do in research. So that is one. And also, sir, as someone who has written so many papers, for a student who is just leaving school, what should publishing a paper be a source of concern? I mean, in terms of visibility. So I just finished my undergrad studies, and I'm waiting for. I'm waiting to know what to do next. Should I go ahead and publish papers? Should I go ahead and start writing papers? Or should I go ahead and intern or volunteer? And if I'm going to be interning or volunteering, should I expect to be paid, or I should just turn to Yakub and manage and not collect money?
Yes, these are very important questions, and I get these questions a lot from students. And my answer for that first question that has to do with interning and doing an internship is not going to be a scientific answer, but it's going to be more of a philosophical answer. And in life, there are stages, and I tell people around me that there are stages in life, and
Mm,
somebody is in 300 level and is saying, "Oh, I need to intern in a place where they're going to pay me so much." Um, don't let me bust the person's bubble, but I'll say that I won't say that if you make money, it's not a bad thing if you make money. But the important thing is that at that stage, what you should be more concerned with is getting these skills that will serve
Mm.
you for life. That's one. Building these connections, very, very important. The life is about connections, human connections. That's the truth. And how are you going to build these connections if you don't go out there and get to meet people, work with them, work for them, understand their needs,
Mm-hmm.
be someone they can think of when or an opportunity comes forth. If
Comes up. Yes, sir.
you understand, if is a leader in the life sciences now sees an opportunity, will he think of your name? Has he met you before? Has anything put you guys together in the same room? How did so?
Mm.
So my answer to students, when students also ask me this, I tell them that if truly you don't have the means, that is maybe the transport fare to be able to go to the interning place, let's say it's really far from where you are, I can understand. I won't ask you to kill yourself. I'll ask you to look for other alternatives. But for an example, let's say you want to intern in UCH, for an example, and because you get the skills, or maybe in Kaba or any of these big places, and if you have the money to go for that internship, I'll tell you, you go, whether they pay you or not. Because one, it puts you in the same room with leaders in life sciences. They know your name,
Yes,
they
sir.
work with, they know your work ethics, they know your name, know your smile. They have an issue, they can say, "Oh, Tomilayo, she has done something for, I know she will fit in here." You won't get that. That is something money cannot buy. And when people say, "Oh, I want to make money," I say, You make money, you'll be tired of making money." Making money itself is not the problem, it's keeping money. So that you're making money is not an issue. It's keeping money and multiplying money that is the problem, not making it. So my answer to students is always, life is in stages. You get,
Yes,
there's
sir.
nothing bad in you making money at 300 level, 400 level when you go for intern. There's nothing bad, but it should not be your focus. At that time, your
Mm.
focus should be getting into the same room with leaders in this field. Look, okay, I'm a lecturer now. How many professors have my number? How many professors know my name? How many professors have I worked with? And if you ask professors I've worked with, what would they say about me? They are the
Mm.
leaders. We are still coming behind them. So these are the questions
Yes,
that
sir.
student and as a researcher must ask yourself. So I think that at this stage, it's not about money for someone who knows what he's doing. It's about learning these skills and getting these contacts. Then the time will come when it will be about money. When you've learned these skills, you've built these contacts, you can now charge the fee
Mm.
that you feel you should charge based on the skills that you've acquired. Now, the second question you asked me was on what now?
Paper visibility,
Yeah,
publishing
yeah,
papers
yeah, publishing.
as an
Yes,
undergrad.
yes, publishing is important, especially for academics. Now, if you are an undergraduate student, I would ask the question I would ask is, what's your plan? Are you planning to go into academia? If you're planning to go into academia, it's very beautiful if you can publish as early as you can, since publishing is all you'll be doing through your life. If you are not going to go into academia, you are interested in going into research and having your own lab, sure, you can also publish. It increases your visibility. It makes people, and the thing with publishing is that it makes people know that you know science, you know what you're doing. Two things that give you that
Wow.
visibility: publishing and then presenting in conferences. Because when you go for conferences, you present, people challenge your work, and then you are able to give the right answers. They know that, oh, Mr. X knows what he's doing. So that is why anybody that is a weak academic, you see that number one, he won't be publishing. Number two, you shy away from conferences, he won't be going, because he knows that these are the places where your worth will be interrogated. And if you are not worth your salt, you'll be found out very, very quickly. So I'll ask, if you are interested in academia, by all means, publish. Talk to your supervisor, tell them, and I can tell you, if you have a supervisor and you tell him or her that you're interested in publishing, 9 out of 10 supervisors would want to publish the work. So maybe you have an odd one or two somewhere that is not interested and is in academia, but 9 out of 10 will want to publish the work. So while it gives you good visibility, if you want to do research, if you want to work in NGO, you want to work in all, it gives you very good visibility. So only maybe the person who is strictly entrepreneurial may not bother too much. Somebody who maybe wants to sell reagents, because even if you have your own lab and it's a diagnostic lab or a research lab, if I want to be sure that you know what you're doing, of course, I need to see your published work. Even though you say you're running a commercial lab, if you have published work.
Sorry, sir. My network kicked me out of uh, of this. All right, sir. All right, sir.
I
So,
think we're
rounding up, what advice do you have for uh, a final advice for students and professionals in the life sciences, either lecturers or entrepreneurs? What advice will you give them concerning how to leverage tech in their work so that they are not behind, and so that they put themselves in a position whereby they can have opportunities, better opportunities within the country or outside the country?
Yes, thank you very much. I just want to say that as a life scientist, researcher, student, we're all scientists, whether you're a student, whatever you are, we're all in the same science boat. Now, it's important, if you ask me what's the number one advice, I'll say it's important that you keep up with the times in which you are. Number one, you must keep up. You can't be left behind. You can't be a scientist today in the life sciences and you don't know what they are talking about when it comes to bioinformatics. You don't know what they are talking about when they are saying they are doing blasts, when they are saying they are doing sequencing. They say they are doing nanopore sequencing. You don't know what they are doing. You must keep up. And it is even more serious today because, me and you started by saying that knowledge, the rate at which knowledge is becoming obsolete is scary. What you knew three
Mm-hmm.
years ago, 4 years ago, makes no meaning today in the world of science and in the world generally. So, as a scientist, as a researcher, you need to keep up and make sure that you are following science blogs, you are following science fairs, you are in different forums and fora, and you are keeping up with the science. That's the truth. Because once you sleep, once you snooze, I'm sorry, you are left behind. And the world in
Mm,
which we are, once you're left behind, it becomes difficult for you to keep up. So every scientist must understand that
exactly.
the world of science is evolving. The world of life sciences is evolving, and you have to do your part to keep up with this very evolving world so that you continue to remain relevant, so that you continue to acquire the skills that make you relevant, that make you somebody in science. If you are left behind with the skills of culturing while people are now doing sequencing, you find out very soon that you are obsolete and you're of no use in the world of science. So that's my advice. Everybody must keep up, stay in fora, stay connected, and make sure that you are not left behind in this tech world. Very important.
Yes, sir. Thank you so much, sir. Thank you for joining me today. Thank you.
Yeah.
It's such a great honor and privilege having you here today. Thank you, sir. I'm
Thank you very
super,
much
super
for having me.
excited to have you here. Thank you, sir. So, thank you so much, Dr. Emeka. Thank you for coming here today. It's such an insightful conversation. I've learned a lot, and I'm sure you've also learned a lot, right? So because we started late, so we are extending a bit of our time today. And do not forget to like, do not forget to subscribe, do not forget to share with your friends and families who are interested in tech in the area of health. So till next month, where we meet again for another edition of Tech in Health, stay blessed. Bye.